Dear students, welcome to the online classes of biology. So, we are studying chapter 9 biomolecules. And in this, I have explained you that we can study the different elements, isn't it? Different biomolecules by different methods. Okay, like chemical analysis can be done. Okay, by grinding the tissues, the living tissues in trichloroacetic acid, isn't it? So, like this, when you do and when you filter the content, you will get two fractions. I told. What are they? The acid soluble fraction and the acid insoluble fraction, isn't it? The acid soluble fraction contains what amino acids, isn't it? Then what the nucleotides, then fatty acids, and then what the this kind of things like what amino acids. Okay, then you will find nucleotides, and then you will also find the type of or smaller type of polysaccharides, that is what monosaccharides, oligosaccharides, etc. are seen in the soluble or the acid soluble fraction. Understood? Among this, I have told you about the amino acids, isn't it? Amino acids we have studied their structure and what their specialities we have studied, and I have also taught you the proteins and okay its structure, etc. Because amino acids are the building blocks of what the proteins. Okay, so amino acids are found in where? In the acid soluble fraction of the what when you did the filtration of the tissues after grinding it in trichloroacetic acid. Understood? So I told these amino acids they are joined to form what? The proteins. So now let's see about another type of molecules, okay, that are found in the acid insoluble and in the acid soluble fraction. So lipids, another group of compounds that are found in water insoluble fraction, are actually what? The lipids, and they are what? Actually they are fatty acids. Fatty acids means what? Fatty acids means a carboxyl group, okay, attached to an R group. Okay, and this R group can be simply a methyl or ethyl or higher number, means the number of carbons can be 1 to 19. For example, palmitic acid. Okay, it's a kind of fatty acid which is having 16 carbon. Understood? So 16 carbon attached to a carboxyl carbon or carboxyl group is what is an example for a fatty acid. Then arachidonic acid. Okay, that is having 20 atoms including carboxyl carbon, means 19 what CH2 group or the 19 carbon groups are there, and which are attached to what? The carboxylic acid, okay, functional group. So what we are studying, I was saying you about what? Lipids, isn't it? So I told you first, I have explained you about amino acids, and along with amino acids I have explained you about the proteins. Now I am saying about the lipids. Okay, so lipids can be what? Or the simple fatty acid. Actually they are the esters of fatty acids with what? The various alcohols. Understood? And the fatty acid means what? Fatty acids are long chained acids. Okay, long chains acid means a COH group will be attached to an R group. R group can be a CH3 or what? Ethyl or methyl or ethyl, or sometimes what? It can be higher, means a number of what? Carbon groups will be attached to the carboxylic acid group. In that way also it can exist. Understood? So example I told what it is. Palmitic acid. How many carbon? 16 carbon including carboxyl carbon. Okay, then arachidonic acid, it is having 20 amino acids, 20 carbon atoms, including the carboxyl carbon. Okay, now fatty acids can be saturated. Saturated fatty acids means what? There won't be any double bond, isn't it? Saturated what the fatty acids, or it can be unsaturated with one or more carbon what bonds. Okay, double bond it can be with one or more carbon double bonds. So that kind of fatty acids are known as what? Unsaturated. If bonds are there, double bonds are there. Saturated means what? Without any double bond. Okay, so a simple lipid is actually what is glycerol. Okay, which is trihydroxypropane. Okay, so many lipids have both glycerol and fatty acids. So you can see fatty acids are found esterified with glycerol. Okay, so glycerol means what? It will have what? 3 OH groups, and this OH groups will be esterified. Esterification reaction you might have studied in chemistry, isn't it? That is the acid and the alcohol group, functional groups, they can react, and they can form what? The ester. So this kind of what ester formation takes place actually during what the for or during the formation of lipids. So when fatty acid, okay, or when a lipid is having both glycerol and fatty acids, okay, sometimes the what lipids are having both glycerol and fatty acids, and in that case, okay, the fatty acids are seen found esterified with glycerol. So that only we have studied now, isn't it? Now they can be monoglycerides if only what are glycerol will have 3 OH groups, okay, and if only one OH group is what esterified with fatty acid, then we call it as monoglycerides, okay, and then it can be diglycerides or it can be triglycerides, okay, and the lipids are also called as fats and oils based on the melting point. Oils are having lower melting point, and that's what they what remain oil in winters. During winters what they remain as oil only, okay, or liquid consistency they keep. Understand? And what if the oil the fats are having high melting point, okay, so they remain as what? Solids in winters. You can see coconut oil and all, isn't it? During the winter season what happens? They become what? They'll become solid. Understood? Then some lipids are having phosphorus, and then we call it as what? Phospholipids. Understood? In the cell membrane we have studied. Then lecithin is one example of that. Okay, then neutral tissues, the tissues in the neural or nervous system, they are having lipids with more complex structures. So lipids, you understood, it is actually found in the acid insoluble form, and so depends upon the molecular size only we are what we can say that the acid soluble what fraction contains water, and the acid insoluble. So even though that is around 800 Dalton, if the molecular weight is there, understood, or less than 800 Dalton molecular weight is there, they will be found in the acid soluble fraction, and those molecules which are having more than more molecular weight than 800 Dalton, they are seen usually in the acid insoluble fraction. But in case of lipids, another feature is there, that is when it is grind, what happens? These lipids will get broken, and they will form vesicles. These vesicles will found in the acid insoluble fraction only, even though they are what molecular weight is less. Okay, they are found in the where only in the acid insoluble fraction when we do the experiment to isolate the biomolecules. So depends on this acid insoluble fractions. Okay, they are having four type of organic compounds, that is proteins that we have already studied, isn't it? Proteins, nucleic acid, polysaccharides and lipids. Okay, so I told the lipids, they are having molecular weight in the range of 10,000 Dalton and above. Okay, and for this reason, okay, biomolecules, that is chemical compounds found in living organisms, are classified into two types. What are they? Those which are having molecular weight less than 1000 Dalton, and they are considered as micromolecules. Okay, and those which are found, okay, and they are found in the acid soluble fraction, and the molecules which are having what more what the weight, molecular weight, they are found in the acid insoluble fraction and are known as what, biomacro molecules. So the molecular fraction or molecular weight, okay, of lipids are actually 800 Dalton, but still they are seen under the insoluble fraction or along with the insoluble fraction, because lipids are what getting, okay, when they are grind they'll form what, they'll form vesicles, understood, and these vesicles are not dissolved or they will not soluble in water, and that's the reason we get it in the where only in the acid insoluble pool. Okay, so by taking the molecular weight, we cannot consider them as macromolecules, but what, as they are not what soluble in water, they will be found in where only, in the acid insoluble pool. Understood? So the acid insoluble pool, I told, they contains lipids. Okay, not because of the molecular weight, because of some other reason. What is it? When they are broken into pieces, okay, they form vesicles, and these are not soluble in water. Understood? So about lipids you understood. Again, in the acid soluble portion, you will find some other compounds. Okay, they are actually carbon compounds with heterocyclic rings. Okay, carbon compounds with heterocyclic rings. So they include what, adenine. Okay, have you heard? Guanine, then cytosine, uracil, and thymine. These are what, nitrogen bases found in the acid soluble fraction. Understood? So when these what nitrogen bases are attached to the sugars, they are known as nucleosides. So what are nucleosides? When nitrogen bases, what are the nitrogen bases? Adenine, guanine, cytosine, then what, uracil and thymine. So these, when they are found attached with the sugar, they are known as nucleosides. Okay, and if a phosphate group is also found esterified to the sugar, then they are known as nucleotides. Okay, and you can see that when this the various nucleotides found in means in organisms are adenosine, guanosine, okay, the nucleosides, adenosine, guanosine, thymidine, uridine and cytidine, okay, are nucleosides. Nucleoside means what, when nitrogen bases are found attached with what, the sugars. Okay, so adenosine is what, adenine plus sugar. Guanosine means guanine plus sugar. Thymidine means thymine plus sugar. Likewise. Okay, and when they are attached to the phosphate groups, they are considered as, or they will be called as what, nucleotides. So the various nucleotides are adenylic acid. So what it will be containing, adenine will be there. Okay, then sugar will be there, and also there will be the phosphate group. Adenosine, okay, sorry, adenyllic acid, thymidyllic acid, guanylyl acid, uridylic acid, and cytidyllic acids are nucleotides. Okay, and these constitute what actually the polymers. Okay, the means the polymers of this are what DNA and RNA. So DNA and RNA are made up of what nucleotides. Okay, so nucleotides contains what? Sugar, then what? The phosphate and a nitrogen base. Depends upon the nitrogen base. It can be what? Adenosine, guanosine, thymidine, uridine, and cytidine. These are what? Nucleosides. And when they are found attached to the phosphate, okay, then we call it as what? Adenylic acid, thymidyllic acid, guanylylic acid, okay, uridylic acid, and cytidyllic acid. Cytidyllic acids. These are examples of what? Nucleotides. And DNA and RNA are formed from this. So, about what now I have explained you about the acid soluble fractions. Okay, what all are the components? So, I have told you amino acids, then what I told you about the nucleotides. Nucleotides, they combine to form what? Nucleic acids. Okay, nucleic acids are formed from nucleotides, or you can say nucleic acids are the what the polymers of nucleotides. Okay, so nucleotide has three distinct compounds. We have already studied that, isn't it? What are they? Sugar, phosphate, and a nitrogen base. Okay, and the nitrogen base is the heterocyclic compound, and the second will be the monosaccharide, that is the sugar, and then a phosphate group. Okay, and the nitrogenous bases found in DNA, okay, are what? Adenine, guanine, cytosine, and thymine. Okay, and the in RNA, instead of thymine, what they are having? Uracil they are having. Okay, adenine and guanine, they are the purines. They are purines, and the rest what? Cytosine, thymine. Okay, all these are known as the pyrimidines. Okay, purines and pyrimidines. So the nucleotides, they join to form what? Nucleic acids, isn't it? Nucleic acids. So this nucleic acids shows what? Secondary structures, and the famous secondary structure of DNA is known as Watson and Crick model. Okay, so according to this model, there are two strands. Okay, two strands are there for DNA. Okay, and these strands will be in opposite direction. You can see one is in the 5 dash 3 dash direction. Okay, and the other is in the 3 dash 5 dash direction. Okay, you can see here this will be in the other direction. Understood? So then the backbone is made up of sugar and phosphate. Okay, and the nitrogen bases, I told the what nucleotide consists of what? A sugar, isn't it? A phosphate group and a the sugar, phosphate, and a nitrogen what molecule, nitrogen what molecule. Understood? Or the nitrogen containing molecule, isn't it? So the adenine, guanine, thymine, then what? Uracil not what? Cytosine. These are the nitrogen bases. So these sugar and phosphate forms the what? The backbone, or the strand is formed of sugar and phosphate, and you can see the nitrogen bases, they are found inside the chain. Okay, they are found or they are projecting perpendicular to the backbone. Understood? And A and G of one strand, they will have what? A pair, adenine and guanine. Okay, sorry, adenine and thymine. Adenine and thymine, they pair with two hydrogen bonds. Okay, and guanine and cytosine, they will pair with three hydrogen bonds. Understood? So the sugar and phosphate, they make the what the strands, and nitrogen bases are found in between them, and adenine and guanine. Okay, sorry, adenine and thymine. They will form two hydrogen bonds, and cytosine and guanine. In between them, there is how many H bonds? Three H bonds. Understood? Then each strand appear like a helical staircase. You have observed now helical staircase, and each step is represented by a pair of bases. Understood? Each step is showing what the pair of nitrogen bases. Okay, and at each step the strand turns 36 degrees centigrade, and one full turn of the helical strand will have what 10 base pairs. Understood? So the pitch would be 34 angstrom. Okay, so the distance between two adjacent turns or in DNA is what 34 angstrom. Okay, then the rise per base will be 3.4 angstrom, and this form of DNA with the this characters are known as bDNA. Understood? So DNA structure, who proposed it? Watson and Crick. According to his what their model of DNA is having a double helix model. Okay, having two what strands which are parallel, anti-parallel to each other. Okay, their direction is different. Then I told you the what the strands are made up of the sugar and phosphate, and in between the nitrogen bases of each nucleotide there exist what hydrogen bonds. A and T they combine with the two hydrogen bonds, and C and G with the three hydrogen bonds. The distance between each step is how much it is? The 3.4 angstrom, and the pitch will be 34 angstrom, and in between a complete turn there is what how many base pairs? 10 base pairs. Okay, so this is the structure of what a bDNA, or the model of DNA proposed by Watson and Crick, is called as what the DNA double helix. So these are the what features of DNA double helix proposed by Watson and Crick. So this much portion is very clear to, I hope. We'll study in the next class. Thank you.